Hello friends, in this video we will learn about DNA sequencing. So many of the sequence method you have heard about is automated DNA sequencing, primer DNA sequencing or Sanger method of DNA sequencing. So in this video we will talk about alternative method. You can divide it into two parts: pyrosequencing, second one is gene chips or microarrays. In this video we will talk about pyrosequencing. In next video we will talk about gene chips or microarrays. So first of all you need to know what does it mean, what's the definition of DNA sequencing. So let's see. It is a pattern that determines the nucleotide sequence in a DNA. So what is DNA? DNA is polynucleotide molecule made up from many single nucleotide molecule. So like assume it is a DNA, many of the nucleotide will be placed on it. So if you anyhow know its a specific sequence, then you might know the cure of many disease, or it can help for biotechnologists as well as other scientists too. So there are many methods. In this video we will talk about only pyrosequencing. Why we call it pyrosequencing? Pyro mean heat or temperature signal. So it is method is based on the principle of determining which one of the four nitrogen bases, adenine, thymine, guanine, cytosine, is incorporated at each step while a new DNA template is synthesized. So it was the definition. Let's see its technique. Okay, as you know, during DNA replication, one of the daughter DNA will be synthesized on parental DNA template like this. So in this process, very first movement, primer will come and start the polymerization. Who will come? Primer will come and start the polymerization process. So first of all, in this many type of deoxynucleotide triphosphate will be attaching, four type of according to their alongly nitrogen bases, DCTP and also DTTP, and you all know their names: deoxyadenine triphosphate, deoxyguanosine triphosphate, deoxycytosine triphosphate, deoxythymidine triphosphate. So four type of deoxynucleotides will be using in it. So as you can see, in very first process, primer will be attaching with DATP in the presence of nucleotidase enzyme. If it will degrade it, and if it is not incorporated or added on our newly synthesis DNA, then it enzyme will degrade it like this. It's like in another DGTP will be adding with primer, and it is also not added on our newly synthesized DNA, then it is also degraded. But in third process, as you can see, primer is attaching with DCTP. So as you can see, it is added, it have added on our newly synthesized DNA. It is newly synthesized, it is parentally DNA. So newly synthesized DNA, cytosine have been added on this DNA part. So now it will not be degraded. Some of the new things have been added on these steps. So let's see what is it. Two inorganic phosphates have been reduced from it, and by the use of sulfurylase enzyme it will give the light signal. If you have get a light signal, it's mean one of the nitrogen bases have been added on our newly synthesized DNA. and so on. The process will be going on again and again up to 200 nucleotides. And again adenine primer is attaching with DATP. Same thing, C have been added. Now the time A will be added, and it will also give the light signal in the presence of sulfurylase enzyme. So as you can see, if any of the nitrogen bases is added, then it is going to give you some light signal that indicate the pyrosequence. G, C, A, G, T, G, C. If these are the nitrogen bases have been added, then every single nitrogen bases have give us light signal. So it is pretty basis. Some of the advantage we should know about it, because it does not require any electrophoresis or any other DNA fragment separation. That's why it is more rapid than another sequence method. Okay, because in this we does not require any electrophoresis or DNA fragment separation, but it is limited. Let's see its limitations. It can detect only up to 200 nucleotides. If you count on the Sanger method, that's more useful for more than 200 nucleotide sequence. So many changes have been made since then. Now automated pyrosequence is also available in the market. So another basic keys for terminology of pyrosequence method: props. What is props? They are synthetic oligonucleotides or complementary DNA part. We call that props. And what is dideoxynucleotide triphosphatase? That is another method for DNA sequencing. We call that dideoxynucleotide triphosphate method, but in this we did not use it, so I will tell you about it in another video. As you can see, I have told you about all the pyrosequencing method, why we call it pyrosequencing. Now in our next video we will talk about gene chips or microarrays. So thanks guys.